So what we're ready to do now is to focus in detail on this equilibrium and to try to answer the question why, under biological conditions, is this equilibrium shifted over in this direction so that the double helical structure is favourable. Now, we've already seen that entropy is not on our side. Okay, so entropy, remember, is this dispersal of energy amongst all the available energy levels. And because that's something that is inherent in nature, a tendency to do that, a state that has a greater range of energy levels over which the energy can be dispersed is likely to be favoured. And we can see that because in the double helix, the freedom of individual atoms to move independently of one another is massively restricted. Okay, because if one of these moves, it drags all the rest of them with it. That puts it at a huge disadvantage compared to this state, where first of all, the two chains are completely separate. So atoms in this chain can move completely independently from ones in that chain, and that gives you a much bigger number of possible energy levels that you can spread the energy over. But even within the chain, as we've seen, because it has now much more freedom to rotate around individual bonds in the backbone, again, that creates a much larger number of accessible energy levels. So entropy is absolutely against this process of forming the double helix. And what we have to focus on is what are those favourable interactions which make it possible for this state to exist. So in order to do that, what we're going to do is to look at the intermolecular forces that are involved in the folding. But what that means specifically is that we're going to look at what intermolecular forces are present in the double helical state, but also we're going to look at the intermolecular forces that are formed in the unfolded state, because it's a comparison of those two that's going to tell us about the position of equilibrium. So the easy trap to fall into is just to focus on the forces present within the double helix and forget that in the unfolded state there are going to be intermolecular forces too. And if we're going to drive the equilibrium over this way to the left, what we're going to require is that somehow the intermolecular forces here are better. In other words, they take it into a lower energy state than the ones that are formed here. So this, remember, is what a single strand of DNA looks like approximately at the displayed... No, try that one again. So this, remember, is what a single strand of DNA looks like at the skeletal formula level. And so we have the backbone running up here, and then the bases up there. And what we're going to do in this section is to focus on the backbone, and then in the next video we'll look at the interactions formed by the side chains. So backbone, remember, has this alternating structure of sugar ring, phosphate, and so on. And the dominant feature that we're going to focus on when it comes to intermolecular forces is the fact that these phosphate groups are negatively charged. So we're just going to examine that a little bit and particularly think about what the consequences of that are going to be for the possibility of folding up of the double helix. The letters DNA stand, of course, for deoxyribonucleic acid. And the reason it's called an acid is because it's a weak acid. And this is the equilibrium that that's talking about. So here you can see that instead of writing the negatively charged phosphate groups, we've written that as an OH. And that's the acid form. But what that's in equilibrium with is the ionized form, where H plus drops off, and then you're left with the negative charge there. So if that's a reversible reaction, which it is, then that means that this is a weak acid compound. And thinking about Le Chatelier's principle, you know that we can shift this equilibrium between the unionized and the ionized form by changing the concentration of H+. Okay, so Le Chatelier tells you that whatever you do to the conditions, it will shift the equilibrium in the direction to oppose the change. And so you know that if you increase the H+ concentration, it will shift the equilibrium to the left. And so we'll get more of the unionized form under conditions where the H+ concentration is high. Conversely, if we decrease the H+ concentration, it will pull the equilibrium over this way to oppose that change, and that will consequently also mean that we get more of the ionized form. And it turns out that the sort of midpoint, where you've got roughly equal amounts of both, is at a pH of around 2. So that's the pH is a measure of the H+ concentration, and it's around pH 2, you're sort of at the midpoint of this equilibrium. So that means that around pH 7, in other words, physiologically relevant pHs, the equilibrium is going to have shifted far over in this direction. So that's why we're going to think of these phosphate groups always as being in this negatively charged state, and we're going to think about the implications of that for folding of the double helix. So a first very simple question we can ask is, if we have a polymer with lots of regularly spaced negative charges like DNA does, so here's just a simplified version of that, if that folds up, is it more likely to fold up like this, so that those negative charges end up facing the outside, or is it going to fold up like this so that they end up packaged on the inside? And just very simple electrostatics tells you that this doesn't look right. Negative charges should repel one another, will repel one another, and therefore it's not immediately obvious that that looks very likely, because you'd think it would just blow up and the negative charges would push one another away and send the equilibrium flying back over in this direction. So for that reason, we should probably expect the negative charges to be on the outside. That's actually backed up by a second consideration, which is thinking about the fact that DNA generally is not folding up in isolation, it's folding up in aqueous solution. So as I say, what we have to think about is what the intermolecular forces are, which are being formed between the negatively charged phosphate groups and whatever is surrounding them, and in the unfolded state, that's going to be the solvent water. And you know, because we looked at this a little bit when we studied ionic structures earlier in the year, that ionic groups can interact very strongly with water by formation of ion dipole bonds. And you know that those bonds are really quite strong, because when ionic substances dissolve, if you take a simple ionic substance, like calcium chloride or something, and dissolve it in water, that's a slightly exothermic process. In other words, when you break down the ionic bonds, which we're always saying are incredibly strong forces, but when you break them down and replace them with interactions between the ions and water, which is what happens when it dissolves, that's actually an exothermic process. In other words, energy is given out, which tells you that it's in a lower energy state when those ions are exposed to water than when they're attracted to each other in the lattice. So in unfolded DNA, those phosphate groups are going to be forming ion dipole forces with water. So when it folds up, if it folds up like this, that's not going to change, because the negative charges are all on the outside, they're still going to be exposed to water, and so to a first approximation, nothing changes in terms of the hydration of those groups. But if it did this and folded up with the negative charges on the inside, then you'd have to strip the water away, and the question is, what would you replace it with? Because you're going to have to find something to interact with those negative groups to compensate for the fact that they no longer have dipolar water molecules to interact with. So both those factors lead you to suspect that a stable structure is going to be much easier to form if the phosphate groups are on the outside. So to see if we're right, let's go back to the actual DNA structure. It's worth emphasizing actually that this isn't just a model that people have built because it kind of looks plausible. This is based on real experimental evidence. In particular, this structure was obtained by X-ray crystallography. What that means is that they crystallized this particular piece of DNA from aqueous solution. And in the crystals, all these DNA molecules are aligned in a very regular way. And if you then fire a beam of X-rays at the crystal, what happens is that the X-rays get diffracted. So as they interact with the electrons in the atoms, they get diverted in different directions, and you get interference patterns as a result of the diffraction from the different atoms in the structure. And from the interference pattern, which you can then detect, you can calculate what the distribution of electron density in the structure must have been. And you can then fit the covalent structure, which you know, to that pattern of electron density and deduce what the structure is. And you can do that, if you get good crystals, to remarkably high resolution. And that's why we're very confident about this structure. So this isn't just something that's sort of been constructed in a biased way because we thought, oh, the phosphate group should be on the outside. There's real evidence that that's the case. So let's look at the backbone, and you can sort of see it already, but we can make it even clearer if we highlight the backbone like this. So if we just select the backbone and then we change the view of it so that it is shown in a space-filling representation. So the base is in the middle, we've left in the ball and stick representation. So here you're just looking at the backbone. And as we rotate it round, you can see very clearly that the backbone is indeed on the outside. And in particular, the phosphate groups, remember the phosphorus is the orange blob, and the red ones around it are the oxygens, which have the negative charge. And you can see they absolutely are facing the outside, just as we predicted. You can actually see that even more clearly if we rotate it round like this, so that we look at it end on. And then you can see very clearly that the bases are all there, in the middle, but the backbone is coiled up around the outside of the structure. So DNA has the sort of structure that we would predict, at least at that level, that whatever it folds into, we would expect the negatively charged groups to remain exposed to the solvent. What you can see in this model, though, quite clearly, is that that doesn't remove the problem, because by folding up into this helix, we bring phosphate groups closer together, not particularly within a strand. So if we look at a particular strand, so if we look, for example, at these adjacent phosphate groups here, even though the DNA is obviously somewhat curled up in order to form the helix, it's not curved so tightly that these phosphate groups are actually any closer together than they would be if it was just in an extended chain. So there's not a problem, the folding doesn't create a problem for the repulsion between these phosphate groups that are adjacent to each other within the chain like that particularly. But what it does do is bring together phosphate groups in adjacent turns of the chain. So between here and here, you can see these two phosphate groups are really quite close together. And that repulsive interaction is a problem. So if we just look at the chain in a bit more detail, what this view also highlights is that the double helix, the two strands don't have a completely regular spacing. So you can see that if we look at it side on like that, that between this turn and this turn, you've got really quite a narrow gap. And you can trace that round the molecule as we rotate it round. You can see that what that actually is a narrow groove which continues all the way along the molecule. So we can follow it there and follow it all the way round like this, and you can see that narrow groove is narrow all the way down. And you call that the minor groove of the DNA structure. Between here and here you've got a much wider gap, and again you can see that wide gap persists all the way round as we rotate the molecule round. So we have there the major groove, and it's the interaction between negatively charged phosphate groups across the minor groove that we're going to really be worried about, because they're obviously going to be quite close together there. The distance actually between two phosphate groups across the minor groove there to there is actually similar to the distance between adjacent phosphate groups in the chain. If it was unfolded, they obviously would be nowhere near one another. So we are increasing the repulsion between the negative charges by folding up into the double helix. So we can add this repulsive electrostatic term that comes from the negatively charged phosphate groups getting closer to one another when the protein folds. We can add that to the loss of entropy as items on our list of things which actually are unhelpful, which are going to tend to oppose formation of the double helix. So we need to think, how do we get away with it? And obviously in part that's because there are other things going on which are favourable, but there are also sort of mitigating factors which make this repulsive term less bad than you might think it would be. The first is again to remind you that this thing really is sitting in a bath of water. It's not sitting there in isolation. And that means that, if you're this phosphate group and this phosphate group, you're not really staring at each other eyeball to eyeball across the minor groove, because in between you've got a whole load of water molecules which will be oriented in such a way that they will help to stabilize the negative charges here and here. So the hydration is hugely important for making this repulsive term less bad than you might think it would be. But there is something else, which is to remember that there must be positive ions in the solution as well, and they can play a role too. In particular, DNA has quite strong affinity for doubly charged positive ions. So magnesium ions especially are often found associated with DNA in various situations. And in fact, when this particular bit of DNA was crystallized to generate this structure, they had, in that case, calcium ions in the solution. And what they found was that some of those actually crystallized in very fixed, regular positions in the structure. And we can see those if we do this. So if we just select the calcium ions and then show them like that, the green blobs are calcium ions. And you can see a couple of very striking things. First of all, if we look at these ones, for example, you can see they're very directly associated with the phosphate groups there. And same thing here. So there's very strong tendency for positive ions to associate around the negative ones on the surface of the DNA. Now, in a crystal, the distribution of ions around the outside is going to be influenced by the fact that really, of course, there'd be other molecules over here and over here, surrounding it in the crystal. So you don't really get a complete picture of the distribution of ions from this. But it certainly gives you a strong hint that the positive ions in the solution are going to play a role in stabilizing these negative charges and allowing them to cope with the fact that they're relatively close together. And also, if we rotate it round, you can see there in the middle of the minor groove is a localized calcium ion. And that too is going to be playing a major role in making it possible for this groove to be stable, even though you've got negative charges on either side of it, because they've got a positive charge in the middle there. And you can see the same thing over here as well. So hydration is important, and positive ions in the solution are also very important in keeping the structure stable. And we can actually find direct experimental evidence of that by looking at how the stability of the double helix changes as the concentration of ions in the solution changes. So here are some data from an experiment where people looked at exactly that thing. So what they did here was simply to take some DNA in solution and heat it up. And what happens is that if you heat it up to a high enough temperature, the two strands fall apart from one another, and the DNA, the word they use in the caption there, is denatures, which means that the folded structure breaks down and you get the separate unfolded chains forming. And you can detect that in various ways, but the simplest one is this. What happens is that the bases in DNA absorb ultraviolet light, in particular with a maximum around 260 nanometers. But it turns out that their ability to do that is affected by their interaction with one another in the folded state. So basically they absorb 260 nanometer light more strongly in the unfolded state than they do in the folded state. And you can see that in these curves. So in this one, for example, it's folded at this temperature, and then as we heat it up, we start to get an unfolding. And so with a midpoint somewhere here, somewhere between 70 and 75 degrees, there you've got an equilibrium between the folded and unfolded states, and eventually by the time we reach 80 degrees, it's all in the unfolded state, so it's absorbing more strongly. And the important thing about this is that they've repeated this experiment in solutions with different concentrations of salt, in this case potassium chloride solution. And you can see that as you go from a low concentration of 0.01 moles per dm cubed up to 1 mole per dm cubed, you get a big change in the thermal stability. So the midpoint is somewhere around 70 degrees at the low concentration, but it's actually not far off 100 degrees by the time you reach 1 molar salt concentration. So what that's showing you is that this ability of the positive ions to effectively screen the negative charges of the phosphate groups from one another makes a huge difference to the stability of the DNA structure. Having this array of negative charges all round the outside of the double helix has some interesting consequences for its properties, one of which is that this structure is quite resistant to bending. Because you can imagine if, you can see it looks fairly linear here. If we tried to sort of bend it round so that instead of being linear like that, it kind of had an arc like this in some way, you can see that what that's going to do is to bring the phosphates on the inside of the curve closer together. And so you're going to get increased repulsion between them, and that's going to raise the energy and make it unfavorable. And you can see that very clearly in this picture. So what they've done here is they've built a model of DNA and forced it to curve round, and then calculated the electrostatic potential around the surface of the molecule. What that means is that if you imagine, for example, a positive charge approaching the molecule from any direction. The electrostatic potential is a measure of what force it feels, as you know, at any point adjacent to the surface, all the way around the molecule. And you can see that the color coding is a reflection of the size of that potential. So red indicates a strong negative potential. And you can see on the outside, because the phosphate groups have been pushed further apart by the bending, the electrostatic potential is actually quite low. Other than directly where the phosphate groups are, you can see the spine of them there, where it's red across the backbone there. But on the outside, overall, the surface doesn't have a high negative potential, but on the inside, it's very red indeed. And what that corresponds to is a strong negative potential at all points on that surface. And basically what that's going to mean is that any negatively charged groups in that region are going to be pushing apart from one another as a result of the repulsion. So we're going to expect that this kind of curvature of DNA is going to be unfavorable because it raises the molecule into a higher energy state. Now that raises quite an interesting problem, which is to do with how you fit the DNA inside a cell. So we need to do a quick calculation here just so that you can appreciate the size of this difficulty that we have. If you measure the distance between two equivalent points on the helix as it repeats its structure all the way along here. So in other words, here is the middle of the minor groove at this point, and then as it bends round, here is the middle of it, where you next see it at the top there. So that distance from the minor groove at this point to the minor groove at that point is about 3.4 nanometers. That's 3.4 times 10 to the minus 9 meters. And if we look at what happens to the chain as it goes through one turn like that to reach an equivalent point in the next turn of the helix, that corresponds to about 10.5 nucleotides. Okay, so as you pass through 10.5 base pairs in the chain, you cover a distance of 3.4 nanometers. Now the significance of that is that, as we've said, there is an enormous number of base pairs in a cell. So the size of the human genome is about 3.1 times 10 to the 9 base pairs. Okay, so 3.1 billion base pairs. And remember that in a cell, you actually have 2 copies of each chromosome. So actually you can double that number and say that you've actually got 6.2 billion base pairs. So if 10.5 base pairs has an effective length of about 3.4 nanometers, we can scale up from 10.5 to 6.2 billion. And a quick back-of-the-envelope calculation tells you that the total length of the DNA stretched out as a linear structure is 2 meters. That's just from one single cell. You have a 2-meter length of DNA. Now the size of a typical cell, if you pick your favorite organ, like say your spleen, a typical spleen cell has a width of about 10 micrometers, so that's 10 to the minus 5 meters. So what we've got to fit in is DNA of total length 2 meters into a cell that's only 10 to the minus 5 meters across. So in other words, the DNA is about 200,000 times too big to fit into the cell unless you curl it up. And we're not just talking about bending it a little bit, we're talking about bending it a huge amount to coil it up so that it will fit inside that cell. And we've seen that left to its own devices, that's going to be a nightmare, because that's going to require a huge amount of energy to bring these negatively charged phosphate groups close together. So how do you achieve it? Well, the answer is that what you do is you wrap it round something which is positively charged, so that it then becomes favourable for all those negatively charged phosphate groups to come close together, because the curling up of the DNA is stabilised by the fact that it brings them all into proximity to positively charged groups. Now, we're not going to get into the detail of this too much, but this picture just summarises this packaging of DNA that happens inside the nucleus. So what's happening in this picture is that as we go down, the magnification is decreasing, so it's like we're stepping further and further back from the structure, so that where we can see the double helix at the top, by the time we're down to here, that just appears as a string, because we've stepped back from it, and that continues as we go down. So when we get down to this level here, we can see the string of DNA, and these individual blobs here are called histones. And they are proteins which are localized within the nucleus of the cell. And they interact with one another to form these blobs, bigger blobs here, which you can see there. And those assemblies of histones are called nucleosomes. And that's the first level of packaging, because you can see what happens is that once you've assembled this assembly of proteins, which is the core particle of the nucleosome, that the DNA can wrap around it. And so you get this kind of structure, which is normally called beads on a string. So there are separate nucleosomes with DNA wrapped around them, and then bits of unpackaged DNA in between. And then the DNA can be packaged further by the nucleosomes interacting with one another to form this higher order structure here, which forms a thicker fibre, and then the fibre itself can curl up into a higher level of structure, and then ultimately you can end up with this most highly packaged form, which is what the chromosomes look like during mitosis. And basically the degree of packaging varies depending where you are in the cell cycle, so that only at this highest level of packaging during mitosis, but it also depends where you are in the genome. So some bits of DNA are packaged most of the time, and then they're inaccessible, so they can't be used to, for proteins to be produced from the sequences they encode. Whereas other bits, the bits in between nucleosomes, for example, are going to be freely accessible, and so those can play an active role, so that the sequences they encode can be transcribed and proteins can be produced. So these higher levels of packaging are basically driven by the protein molecules interacting with one another at various levels. But what we're going to look at just is at this first level, because this is where the DNA has to wrap around these protein assemblies, the nucleosome core particles. And we're just going to have a look at that in more detail, because that's what's relevant to what we've been talking about, which is how do you bend DNA in the cell. So this is the crystal structure of the nucleosome core particle with DNA wrapped around it. And again, what they've generated here is an electrostatic potential surface. So remember, blue corresponds to regions of positive potential, and red corresponds to regions of negative potential. And you can see round the outside of this core particle, it's overwhelmingly blue. And that's what makes it possible to coil the DNA around it, because all these negatively charged phosphate groups, even though we're forcing them to come closer together by bending the DNA in this way, that's overridden by the fact that it brings them into close proximity to these positively charged protein side chains, which are strategically located all the way around the outside of this particle and allow the DNA to bend in that way. And in making biology work, there's a huge amount of interaction between DNA and proteins involved in, all kinds of processes, like, for example, the expression of genes to produce proteins. And always the proteins that do that, often they need to recognize specific structures, and they may do that by interacting directly with the bases in the middle of the DNA. But nearly always they make use of this electrostatic attraction between positive charges on the protein and the negatively charged phosphate groups to anchor the DNA in place. So the phosphate groups have this important functional role, as well as just being a sort of nuisance when it comes to trying to fold up into the double helix. So we've seen how the molecule is able to cope with that, but what we've also seen very clearly is that those phosphate groups, the folding of the backbone, is not going to drive formation of the double helix. It's not a favourable process. It's something unfavourable which we can cope with these various strategies that we've talked about, but it's certainly not going to drive the folding. To understand that, we're going to have to look at the interactions, the intermolecular forces formed by the bases, and that's where we'll go in the next video.